introduction of the LungMAP and the LungMAP.net, which is the central portal. So I will focus on some of the community portals and we developed in recent years, and how we use those to understand the lung in both healthy and disease conditions. So the first thing I want to introduce you is the LungMAP cell ref. I don't know any of you doing the single cell yourself. If you do, you will agree that the accurate cell type identification is still the key and rate limiting step, because everything is built on the top of that. So the previous studies are mostly using individual single cell reference. So basically, if it's a high impact paper, or the markers you learn from the paper, and you identify your cell types. And for this approach, it's often leading to the missing cell types because each individual study will be sampling different, right? And also the reproducibility is another issue, and a lack of common cell type nomenclatures. And Nathan just introduced the cell cards, which indeed represents a rigorous, lung cell catalog with standardized cell names and associated markers. So this basically motivated us to do a guided construction of a cell reference for human and mouse using cell cards as a dictionary to promote the automation and the standardization of the cell type annotation. So Dr. Minze Guo from our group, he developed a novel pipeline to do this, and for ensure the comprehensive design, he collected the data from both published and unpublished domain to cover the lung from different region, and both male and female, age span from 10 to 80, and technology speaking, we have 10x, 3 pound, 5 pound, single cell, single nuke, etc. So through the integration and remove the batch, and we did the strategy using both supervised learning and unbiased clustering. Supervised is using the basically rank the cell based on their expression on the cell card signatures. And unsupervised is to ensure we have the discover ability to find the cells that are not yet in the dictionary. So using this strategy, we constructed the cell ref for both human and a mouse. Human has 48 cell type, and the mouse has 40 cell type. And mouse is more developmental, and human is more in the adult stage. So we actually did extensive benchmark and evaluation to ensure the cell ref, the accuracy and the reliability using multiple metrics statistical models, including the one developed by the Dr. Nathan Salomonis lab, single cell triangulate. And the take-home message is, so basically the cell ref prediction is highly accurate and stable. So we tested the cell ref utility using 8 independent testing data sets from both normal and disease. Here is one example that cell ref can accurately predict normal lung cell types. And the left panel is the original publication, the cell annotation. The middle is the cell ref annotation. And we measure the performance using multiple metrics, including the F1 score, precision, recall, accuracy, and Matthews correlation coefficient. Bottom line is you can see all the based on all the measurement, we have outstanding performance. So highly consistent with the original annotation. So many of you actually interested and ask if we can use the cell ref to identify disease cell types. So we tested this utility in using two lung disease. One is based on the previous publication from our own group, and that's using a rare lung disease called the lymphangioliomyomatosis. And there we identified a cluster of cells that are uniquely, expressed in LAM that not similar to any of the endogenous lung cells, but share the transcriptome similarity with the uteromycete. So there we actually predicted the uterus as the likely, origin of the LAM cell. So here we using the cell refs prediction compared with the original annotation. You can see the LAM call cannot map to any of the normal cells in the cell ref, and with the lower is the prediction score showing here, and like far away from the cutoff line to be considered as confidently mapped. So the second case we use are IPF single cell data sets. Also, there they identify a previous unrecognized cell type called keratin 5 negative and keratin 17 positive. They think it's a pathogenic cell and enriched in the IPF lung. So here we did the comparison. You can see this cell group as the lower is the prediction score. Again, far away from the cutoff line, and indicate we have no confidence to map it to any of the normal cell type, and suggest it may be a pathogenic cell type. So Nathan mentioned that the asthma's incidence also is hosted in the LungMAP.net. And so if you think the cell ref is useful, and you can use this online tool, which enable you to load your own sequencing and follow the simple steps. You can do the QC, UMAP, cell plot, and download your results. And for advanced user with large data sets, the website may be not able to handle that much data. Then we also provide all the code, step-by code that is freely available in the GitHub. So for most of the user, actually, after I talk with them, they like to use the CellRef as a daily, operating resource to check their genes and check their cell types. And for that, we developed multiple, interactive web tools for search and display function. And Nathan mentioned the Shining Cell and hosted on the LungMAP.net, and I can show you more of the slides of the functionality that you can use the LGEA to do the similar search. And so LGEA is a web portal that developed and hosted in my lab, and just show you the major timeline and major version release. And we start building that in 2014, which is the first year of LungMAP phase 1. So it's a LungMAP baby. So initially only have 2 single cell data sets there. And you can see with the time, we have more data, more functionalities incorporated into the LGEA. And the version 3, we released Lung at a Glance tool sets, which include the region at a glance, and a cell at a glance, and a gene at a glance. And then this year, we incorporated the cell ref and build the interactive connections with the tools in the LungMAP. And Inadu is our previous webmaster. Our goal is always to build user-friendly, intuitive, self-explanatory cell portal for maximum usage. And the technical objective for us is to establish bridge to connect the lung ontology and the imaging and the corresponding omics data together. So Jihong is our new web developer, and this is the new interface for the LungMAP phase 2. And you can search for both, mouse or human. You can click the region at a glance, cell at a glance, and a gene at a glance. And the cell ref is here. So just for example, Dr. Morris' lab published a paper on Nature last year, and they identify a new cell type they call the RAS, which is a respiratory airway secretory cell. And if you like to find more of the gene expression or cell pattern in the cell ref, you can search RAS under the cell info, and it will. This is the cell ref UMAP, and then we extracted this the RAS cluster here. And if you click the cell, it will invoke the cell at a glance at LGA, and they give you the definition and citation and positive marker, surface marker, etc. And we also updated the ontology tree to include all the cell type that described in the cell ref. So we also provide interactive connections from cell ref to cell cart, and Nathan just showed you many of the nice comprehensive features in the cell cart. So user can always from here to jump to the cell cart to get more information. And in addition to the top rank the markers, if you want to see more markers, you can click here to will give you an interactive table to list all the differentially expressed genes in the RAS cluster. And if this is still not enough, you can have the flexibility to type any gene in your mind if you type the gene, it will return you the gene expression patterns and the statistics. So the LungMAP Center, at least the RF Center, is more and more towards the disease and building the resource to understand the disease. Here is just an example. We recently built the LAM cell atlas. Currently, the major function included the LAM transcriptome, epigenome, connectome, and the project, and the LAM imaging. Transcriptome contains the single-cell RNA-seq data from the uterus, lung, and the kidney. And the epigenome mostly currently is the single nucleotide-seq. And connectome basically is the cell-cell interaction through the ligand receptor network. The project actually designed as a story page to introduce the recent studies, single-cell omics studies related with LAM. And the imaging is collected for the histology and immunofluorescent, confocal imaging, etc., related with the LAM study. And this year, we just finished a single-cell multi-omics study for the alveolar capillary dysplasia, ACD, which is another rare lung disease. And the paper is currently in press by Blue Journal. But we already have the data published in LGA. We always release our data before the publication, so everyone else can use it to build their own hypothesis. So given the time, I don't think I can cover all the functionalities. But if you are interested, you can go to the LGA. Under the homepage, we have the help, and you can, under the help, we have a series of tutorials, and those tutorials actually build based on users' inquiry and on their frequent asked questions. So hopefully you found the scenario also suitable for your case. And again, just like Nathan said, you can always email me and ask for more specific questions how to use the LGA or any of our tools. So yes, hopefully I can convince you to use LungMAP as a living resource for your study. I listed some of the URL here, but I think it's to me, it's more easy just to Google LGA or LungMAP.net, and all those links will come out. So I think, yeah. So last, just wanted to give the acknowledgement to the people involved in the work, and the people directly involved in today's talk include Dr. Minze Guo, Ina, and Jihong, and everyone in my group, UC colleagues, and the LungMAP colleagues from CCHNC, DCC, and other research centers, and all the members, and also the funding mechanisms. Thank you, and feel free to ask any questions now or later.
Hey, Yan, I was just I was curious because I haven't seen a lot on this lately, but how good are the latest generation of organoid models at recapitulating disease level single cell gene expression phenotypes? Are they capturing the major cellular heterogeneity and molecular heterogeneity, or is that not yet there yet?
I think it's not complete, definitely not there yet. And actually, Nicole probably can answer better, because Minxia's lab actually is doing some cutting edge organelle stuff, and we are have some of the collaboration through the Cedars-Sinai Medical Center. They try to put the lung into the 3D chip, and mostly focus on the mucosa area and the air-liquid interface. And we did some comparison, and it seems like they can differentiate into some of the major cell types, not all the type, but and also they seeing for example, the cilia beat and the CCFTR channel, all those. So it's getting it definitely hold the promising. And Minxia, I think their group doing a lot of like a more disease model, and so ACD, BPD, yeah.
I actually do have a question, just kind of like bouncing off Nathan's question. So speaking of organoid disease models, are you aware if anyone in within or outside of the LungMAP group is working on modeling LEM using organoid systems?
Not really. That's the thing I wanted to develop the collaboration with your lab. I really looking forward for that. But it's tricky is there's no funding to get, and this is a very and I try to find a way, like maybe some pilot funding we can working together. And I am totally a dry person, but this is super exciting. I think the direction. Yeah, we don't have currently by the the whole LEM field, there's no successful organelle yet. So that's cutting edge stuff. Yeah.
Well, okay, yeah, that's really exciting. We can definitely explore that. And I was just excited at the prospect of doing that, and then performing a bunch of like single cell multi-omic like studies, and then have that be in the LEM cell atlas as well. I think that would be really cool. I actually just also have another really sort of simple like question, kind of out of curiosity, for those imaging data that you've shown sort of in the LGEA portal. So those, are they from existing like publications, or they are deposited by users?
Yeah, so the Almira Nathan mentioned, that's it contains more data. For the LGA, mostly directly is connected with the lung imaging from which hosted in Dr. Wissa's group. I think it's still like more than 2,000 imaging gallery from the both mouse and the lung, and mouse lung, human lung, and primate lung. Yeah.
Okay. And I'm not actually familiar with any other major lung image repositories, because it'd be great to think about including those, but I don't know if they exist. Do they?
Yeah. Yeah, I think a lot of people using the lung imaging. I think UCSD. There are other. They just said, can we use that for a supplementary table, right, guys? Yeah, as long as Dr. West says agree, I'm fine. Yeah, because that's all developed. The imaging is generated in his lab. Yeah. Yeah, I don't think the Human Cell Atlas has the imaging part.
Or I'm just even not like no other lung efforts. It seems like that's not really a focus.
Right. Yeah. Yeah. This is a big, like a unique thing for LungMAP, I think.
A question I can't ask in LungMAP is how what because it's NCI related. And it's all, you can't talk about cancer with NHLBI, but like what are predominantly in most like lung adenocarcinoma, there's if you talk about like the major lung cancers, so there's lung adenocarcinoma, there's I'm actually not familiar with other major
small lung
lung.
Yeah.
Are they, do they tend to be, is it what are the cell types that typically gain the pre-malignant mutations that undergo clonal evolution and ultimately expand? Is it fairly well known, or is that in terms of what and is that process fairly well understood?
I'm not the best person to ask that, and I think Catherine is actually did previously on the mouse model, and also so the small what they say I think
and I know Vlad probably and his wife knew, but yeah, they're now gone.
They actually, so the lung cancer mostly Catherine's lab doing that, and also the guy, the Japanese, the Yutaka, from
okay. Okay.
So he is doing the adenocarcinoma using in the study the NCAX 2.1, the role of the NCAX 2.1 regulating the
Okay, that's great to know.
I never did any of those myself.
No, thanks so much.
It's awesome. Thank you again for taking time off for a busy schedule to help me with this webinar. I think I definitely learned a lot. So yeah, thank you again for the attendees for joining us again. And if you have any questions related to LungMAP, definitely feel free to reach out to Dr. Shi or Dr. Salomonis. I think they'll be very happy to answer any questions. So yeah, thank you again, and yeah, have a good rest of your day.
You too. Bye-bye.
Bye.